Hi everyone, my name is Lara Gardiner and today I'm going to be presenting on the circadian rhythms of fruit flies, and I just want to give a special shout out to my advisor, Dr. Kyle Gabrogi, for supporting me during this experiment. So I'm going to start off on a pretty somber note by stating, as of 2020, 8.4% of all adults in the United States experienced a major depressive episode. And for this population of individuals, there's a wide range of therapy options available. However, most of them are very costly, they're time-consuming, and they're oftentimes limited to those with health insurance. So this is where bright light therapy comes in. Bright light therapy is a new therapy that's meant to ease symptoms of depression. And essentially, what the bright light therapy does is lengthens an individual's photoperiod. And a photoperiod is an individual's daily exposure to light. And lengthening this photoperiod has been found to ease symptoms of major depression. And although this seems like a very good therapy option for individuals suffering from depression, there are a few details about this bright light therapy that have yet to been investigated. These details include the specific color temperature of the light that would be able to best alleviate the symptoms of depression we're talking about. So for example, is it this warmer tone of light that you see on the left, or is it a cooler tone of light that you see on the right? Which one of these lights would be able to optimize the benefits of this light for the people suffering from depression? Another gap in the literature would be which specific symptoms of depression are alleviated through this mode of therapy. So for example, if an individual suffers from appetite-related symptoms of depression, however, the bright light therapy is only able to alleviate sleep-related symptoms of depression, it's really important that we don't prescribe this kind of light to them because it won't have any benefits for them. So these few details need to be further investigated so that we can ensure that we're optimizing this therapy for the people who need it. So this is where my project comes in. And essentially what I did was I looked at two different light conditions, 2,700 K light and 6,500 K light, and I looked at how they were best able to alleviate sleep-related symptoms of depression. And I did this through measuring circadian rhythms. And so the first step to my experiment was deciding which animal model to work on. And because of the available genetic tools, my previous experience, the short lifespan, and also the overlap with human brains, I decided to work on the fruit fly. It's really surprising to think how similar our brains are to the fruit fly, but there's actually a lot more overlap than you think. And so after deciding that I was going to work on the fruit fly, my first step was using one of those genetic manipulation tools to essentially silence all serotonin neurons in the fruit fly's brain. And so this is a really important step of the experiment because essentially one of the common phenotypes of individuals with depression is that they have suboptimal levels of serotonin. And so it was really important that I silenced all the serotonins in the fruit fly's brain so that I could kind of mimic an individual with depression. So after doing this, what I did was then I had to monitor their activity over 24 hours, also known as their circadian rhythms. And I did this using the Drosophila activity monitor. So essentially what happens here is that each individual fly is housed in its own individual tube with food on one end and a cotton plug on the other end to allow it to breathe. Then what happens is this tube is clicked into the activity monitor, and each activity monitor can house up to 32 different tubes. So we can essentially track the activity of 32 different flies at once. And so how the activity is actually tracked is that through each tube there are many infrared laser beams going through the tube. So for example, if I'm shining a light and somebody puts a laser beam and somebody puts their hand in front of it, I'm going to know that there was movement in front of that laser beam because the beam was broken. So essentially what happens is if the fly moves in front of a laser beam, that beam break is tracked by the computer as movement. So after getting movement of these fruit flies for over 24 hours, I essentially had to plot the data that I received. And so here we can see a comparison between a wild-type fruit fly and a serotonin knockout fruit fly. And the wild-type fruit fly essentially just represents a fruit fly that has normal levels of serotonin, whereas the serotonin knockout fruit fly represents a fruit fly that has suboptimal or no levels of serotonin whatsoever. And on the y-axis, you can see the activity, which is counts, and that's essentially the beam breaks that I was just referring to, how many beam breaks occurred in each hour slot. And then at the bottom, there's the Zeitgeber time. Essentially, the Zeitgeber time is just the 24 hours in a day, where Zeitgeber time 0 represents when the lights came on in the chamber that the fruit flies were housed in, and Zeitgeber time 12 represents when the lights turned off in the chamber that the fruit flies were housed in, essentially giving the fruit flies a 12-hour light, 12-hour dark cycle. And so what we see here is pretty representative of the circadian rhythm of a fruit fly. There's a bout of activity in the morning in anticipation of the lights coming on, and then a bout of activity in the evening in anticipation of the lights coming off. And so essentially, what you can see here is that the serotonin knockout flies experience greater morning activity, whereas the wild-type flies experience greater evening activity. And it's really important to establish this difference in these two graphs because essentially we had to make sure that serotonin was playing a role in regulating the circadian rhythms of the fruit flies so that we can continue with the experiment. So the next step is actually exposing them to the two different light conditions. And essentially what I did is I took one group of serotonin knockout flies and exposed them to 2,700 K light during that 12 hours of light a day, and one group of flies and exposed them to that 6,500 K light, which is like a brighter light, and the 2,700 is like a warmer light. And I exposed them to the 6,500 K light during its 12 hours of light a day and received these two graphs. So essentially what we can see here is quite a big difference. That's the first important thing we have to note, is that there's a big difference between these graphs and the one I just showed you, because these obviously show much more greater morning activity. And that's important because then we know that the color temperature of the light is actually doing some sort of regulation of the circadian rhythms. There's some effect that the light temperature is actually having. And so here we can see for the 6,500 K exposure, which is that bright, cool light, we can see that there's actually greater morning activity in comparison to the 2,700 K exposure. But the 2,700 K exposure has greater evening activity. And so although this may not provide precise and conclusive evidence as to which exact color temperature is best able to alleviate sleep-related symptoms of depression, what we can see is that color temperature does actually play a role in somehow regulating these circadian rhythms, and that it's important to do future research so that we can start specifying conditions like these so that we can optimize this bright light therapy for individuals suffering from depression. That's it. Thank you.
Thank you so much. That was awesome. I had two main questions.
Okay.
One was, did you, what temperature was that light in that first set of graphs where you just compared wild type versus serotonin knockout?
So that temperature was just like a regular baseline light. It wasn't anything like the 2,700 K or the 6,500 K. It was more so like one in the middle.
Okay, yeah.
As you can see in this diagram here. Whereas you can see that the 6,500 K light has like that blue tint, whereas the 2,700 K, sorry, the font is kind of small, has like a warmer tint to the light.
Awesome, thank you. And then I was wondering,
sorry.
Are there any other, is there any other evidence in the literature of other effects to serotonin knockout, globally in the brain? Because I'm guessing that human depression is a little more specific than just
right. So,
serotonin.
Like I said, humans experiencing depression often have suboptimal levels of serotonin, which is said to, it's called the serotonin hypothesis. It's said to cause that depressive mood and also a lot of the other symptoms that are listed here, for example, changes in appetite and things like that, because serotonin is more just kind of like an emotional regulator, and that's why it has those deep effects. But there is some literature that pointed to the fact that fruit flies, when they do have serotonin knockout, they do exhibit a depression-like state, and that's why in an attempt to replicate or I guess mimic an individual with depression, I decided to knock out the serotonin neurons in the fruit fly brain.
Can you talk a little bit more about the similarities and differences between a fruit fly's brain and our brain?
Yeah, okay, so there's similar genes involved. So for example, the fruit flies have these areas in the brain called the mushroom bodies, which house the serotonin neurons. So it's really surprising, but this, for example, fruit flies have similar neurons in the brain. For example, we both have serotonin neurons, both have dopamine neurons, and I can't remember the number specifically, but there's something like, I think it's around 28 of the same genes that regulate human brains that are also in fruit fly brains. So for a long time now, I think over 100, maybe not 100 years, but fruit flies have been used as an animal model for neuroscience research because they're just so similar to humans. And they have a short lifespan, like I discussed.
Really, really interesting. I want to know more about Nathan's question too, but I'm coming back to the human part of this. I wonder if there have been studies in the natural light world in terms of the effects of these different kinds of color temperature scales, where they exist actually geographically, and whether there have been studies on humans and the effect of even seasonally or perhaps in equatorial regions, what's going on there, and how you take those studies and do something with them in terms of informing your own research.
Yeah, so the color temperature of the sun, I'm pretty sure it's above 6,000 K, so it's a very, very bright light. And that's what I was trying to replicate by using such a bright temperature light. I was trying to replicate something like having the sunlight on your face. At first, I wanted to actually expose the flies to sun, but I was like, how was that? It's kind of hard to like put this activity monitor somewhere in the sun where I can regulate the clouds and all of that. So it's very difficult to control that. So I tried to replicate the sun through this. But yeah, like I said, earlier in the presentation, essentially lengthening individuals' photo period, which is your daily exposure to light and sunlight, is meant to alleviate symptoms of depression. So in a way, there was a lot of influence as to sunlight and how that plays a role in this experiment. And you can see how that plays into things like seasonal depression and things like that, because obviously your daily exposure to light decreases. Thank you.